my institution relies on the work of about 1,000 qualified workers in the field of research, development, production, quality control, and manufacturing of vaccine, as well as some modern facilities where research and production are carried out using the most rigorous requirements. I want to highlight that most vaccines used in our national expanding programs of immunization are produced by the National Biotechnological and Biopharmaceutical Institution, mainly Finland Institute and the Center of Genetic Engineering and Biotechnology. This is our leader product, Bamengo. Bamengo was the only licensed vaccine against Serotype B meningococcus for many years and got the gold medal given by the World Intellectual Property Organization in 1999. And after 60 million doses has been applied, it has proved to be safe and effective. This is a bivalent vaccine of capsular polysaccharide of Neisseria meningitidis serotype C and outer membrane vesicles of Serotype B meningococcus, with many immunogenic and different proteins. The development of this vaccine started in 1983 after a huge epidemic of meningococcal disease with predominance of Serotype B. The development of this vaccine finished with a large phase 3 clinical trial with an efficacy of about 83%. This vaccine was applied in 2 stages. During the first stage, a mass vaccination campaign in people between 3 months and 24 years of age, and during the second stage was included in the National Immunization Program. See the sharp drop of the incidence rates in all age groups after vaccination. Last year was only 0.1. Bamengo is widely used in Latin America, where more than 25 million people have been vaccinated. For example, in Brazil, where the fall-off of the incidence rate was also proved after vaccination. As well as in Uruguay, see that the post-vaccination incidence rate, of course 0, was even lower than the pre-epidemic incidence rate. We can say that Bamengo is an effective vaccine against meningococcal disease B and C serogroup in infants, children, and adults. But on the other hand, it's important to say that the outer membrane vesicle strategy is being used for the development of other meningococcal, Shigella, and pertussis vaccines. This is other of our vaccines, Vaxpiral. Vaxpiral is a leptospirosis vaccine, the only one in the market, and this vaccine has controlled the incidence of the disease in our country. After a large phase 3 clinical trial, the serotype-specific efficacy was 78.1%. And more than 90 million doses has been applied with an excellent profile of safety and efficacy. This is other of our vaccines. VaxTB, vaccines against Salmonella typhi, and is supplied to the National Immunization Program, substituting the former wholesale vaccine. Many different clinical trials have been carried out using typhin B as vaccine control, and the reactogenicity and immunogenicity of both vaccines were similar. It was included in the Cuban immunization program. About 3 million doses has been applied, and adverse event has been infrequent and mild. Vaxted, a highly purified tetanus vaccine, has been used in Cuba since 1995, and it's important to highlight that this vaccine, this antigen, is also used for the conjugation of the new Haemophilus influenzae type B Cuban vaccine. Other common vaccine used in the expanded program of vaccination are produced also in our institution. And DDP is used in the formulation of the neoadjuvant tetravalent and pentavalent vaccines, whose final production is in the Center of Genetic Engineering and Biotechnology. This is a summary of the medical registration application worldwide, seen that mainly in Latin America and also in Asia, including Syria and Pakistan. Well, the institutional pipeline, the research project, basic research projects, the candidate vaccines in development phase, and I have already talked about the vaccines in production. I'll show you in brief the main basic research projects. Inactivate Vibrio cholera host cell vaccines in tablets or microencapsulated is in the preclinical phase, and this is a collaborative research with some Spanish institutions. A tuberculosis project that is addressed not only to the development of vaccine, but also diagnostic kits. And these are the main strategies of TB vaccine according to the point of view of our institution. The expression of Mycobacterium tuberculosis antigens in leaf attenuated vector, the development of DNA vaccines, recombinant subunit vaccines, and the use of Mycobacterium Havana strain. This is collaborative research with the University of Science of Malaysia. The development of vaccine adjuvant is other of one of the most important projects. You know that aluminum hydroxide is not a good immunoenhancer. That's why other adjuvants are being developed. Like Neisseria meningitis protoliposome is an excellent adjuvant that induces a cellular T helper 1 pattern, and is the main adjuvant of human cancer vaccines. Also, is the raw material for coagulate, other excellent adjuvant that enhance the immunological memory and induce also T helper 1 pattern and mucosal immunity against homologous and heterologous antigens. Well, coagulate is being studied as a proper adjuvant for mucosal vaccines, and also as vaccine adjuvant for therapeutic or prophylactic vaccines against malaria and leishmania. We'll talk in brief something about diarrheal disease. You know that vaccines against typhoid fever, cholera, or rotavirus are available. However, shigellosis is still a big challenge. That's why we are developing a combined formulation of Shigella protoliposome with Salmonella typhi polysaccharide. And a new approach for obtaining a Bordetella pertussis acellular vaccine that is based on the outer membrane vesicle technology. Finland Institute also obtained monoclonal antibodies to support the development of vaccines and also for diagnostic kits. For instance, monoclonal antibodies against relevant antigens of Neisseria meningitis, Vibrio cholera, and Mycobacterium tuberculosis. And finally, the basic project or fundamental projects, the development of experimental animal models for immunogenicity studies, active and passive protection tests, and for preclinical toxicology. And now, I'd like to show the main projects in development. A genetically live oral cholera vaccine, multivalent pneumococcal vaccine, and meningococcal vaccine, both based on halal technology.